Okay, so first, thank you very much. It's a real pleasure to be here, have the opportunity to give this talk. So I'm going to talk about particular constructions made of DNA, which are really well structured. These are called DNA origamis. I want to discuss how they are formed and how they can interact with lipid membranes. So to start with, let me motivate a bit this talk. So these are pictures. This is a model of a structure that has been synthesized and characterized in a group in Montpellier, in France. The group is led by Gaëtan Belot. So the structure is this thing over here. You can see the typical size here is 100 nanometers. It has two legs that allow this structure to land on top of a bilayer. Has a central part here, which is a piston that can slide and can exert a force on a protein which is embedded in the bilayer. So I had the chance to participate a little bit in this work, modeling the force that can be exerted by this nanomachine. Let me show you EM images that show that this is not just a model. This works, and after that, there is a full characterization of the action of pulling on a protein, which is actually an integrin, and how this can be characterized goes really beyond my talk. So here the idea is that you just take DNA and you can manage to do a nanomachine, let's call it this way, that can have well-determined interactions with cells, in which was the case here. And well, this is, for me, this is very interesting. It's like playing biology, but not having the background of biology, right? Doing that with DNA. Okay, I'm not going to talk about these really funny structures, not as funny as these ones. I'm going to focus on how these structures are made. I should mention that the first paper that developed this technique of DNA origamis is by Paul Rosborn in 2008. So it's 17 years ago, and the number of models that explain how the formation of these structures goes are very few. So I'm going to focus on this. So let me just tell you how the formation of this origamis goes. So you have this red line, which is a very long single-stranded DNA, so very floppy structure. And this is, at the end, you want to get some particular structures, let's say this rectangle or this constraint rectangle. So to do that, you need other agents. These are called staples. These are these short sequences of also single-stranded DNA. And to get to this structure, you need to compute the interactions between the staples on what is called the scaffold. And basically, what the staples are doing are they want to hold together, like here, two parts of the scaffold that are really separate in sequence. Okay, so the experimental process is very simple. So first, do the computation, right? Decide what structure you want. Then do heat all the solution. Everything goes in solution. So heating is to avoid any secondary structure that could appear in the scaffold. This is very long, so you expect a lot of secondary structures, and they slowly anneal and expect to get this thing. Now, when you think, so DNA is made of 4 letters. So the scaffold has 7,000 length. The number of possible repeats in this structure is very, very huge. When you decide to do this computation, what you are somewhere is praying that everything that is not included in this model is not going to play a role, right? And it seems to work. I mean, this has been going on for 17 years. I just want to convince you that there is something, a trick which is hidden there. Okay, and for that, I need some modeling. So I want to discuss three simple questions. So the first, as I said, is how the folding process goes. The second question is, are there conditions where this folding doesn't happen or very badly? And if I have time, at the end, I will discuss the interaction with lipid membranes. Okay, so to describe the folding, I'm going to use a very simple method, which is UV absorption. Just a quick reminder. So everything is based on the fact that absorption of unpaired bases is higher than the absorption of paired bases, so that if this is temperature and you have this double helix, if you follow the orange curve, when you hit the structure, you expect it to unfold, so the absorption is going to increase. But if you destabilize a little bit, like adding a loop, like here, the curve is going to be shifted to the left because it is a bit less stable. And actually what is done is represented is not this curve, but the derivative of the curve against a temperature. So for each structure, you have a typical peak, which is centered about what is called the melting temperature. Okay, so now let's try to model. Let's decide that we want to study this rectangle. And let's try to model how this happens. So you know that the thermodynamics of DNA has been really well established since the late 90s. And the very first naive model you would like to try on this structure is, well, locally we have a lot of double helices. Let's say that all of them are independent, and let's put these thermodynamics inside and see what happens. So here you have three different rectangles, three different origamis. The number of staples is mostly the same, and these are the the annealing curves, the UV absorption curves that you get as a function of temperature. And keep in mind that in order of increasing stability is red, green, blue. Okay, now, if you apply the independent model of helices, what you get is red, green, blue, but in the other direction, okay? So it doesn't work. Now, if you think a little bit, I said that the staples are there to hold together different parts of the scaffold, and in doing so, you are obliged to create loops, okay? Okay, loops are also well characterized in this thermodynamics. The penalty goes like log of the length of the loop. But what happens here is that eventually you can get staples that are bound to the loop, okay? So the length of the loop is dependent on what happens outside the loop. So this is a bit convoluted, and we decided first to study a very, very simple structure. Is this kind of, so we have two staples, B1, B2, and we have a scaffold which is the complementary of this. And to make things very simple, we decided to have very different compositions for on the two staples. One is only composed of A and T's, and the other is much more stable, is composed only of G and C. Okay, so let's see what happens here. Let me guide you through this picture. So the easiest thing to understand is the position of this peak over here. This is just a double helix. Now let's put this staple, the red, in this position. So this is just a loop, formation of a loop, and this is destabilized. But what happens if in solution you also add the GC rich staple, this one, the curve is shifted to the right, okay? So that means that you cannot only take into account this loop, you need to take into account the fact that other staples can bind to the loop. This is what we call cooperativity. So the folding of a less fluctuation structure is easier in the presence of other staples. And let me give you another example. So this is a standard loop. This position is what we call the inner loop. The inner loop is not parameterized in the usual thermodynamics of DNA. And you can see that it is the more unstable structure we have in here. But again, if we add the other staple, the GC rich staple, the effect is the same. We stabilize everything. Okay, so this was the very simple structure. So we did a parameterization of these effects as a function of the composition of different staples, always in this context of 3 staples. And what we decided is to include all this information in a algorithm, which is iterative, in which we try to compute the probability of binding of a staple as a function of temperature, taking into account what we know of the other staples, okay? So we have this set of reactions, a staple plus a environment of the staple goes to the binding of the staple. To solve this law of mass action equations, we need the thermodynamics. And as I said, what we did is start from what is known. So this Santa Lucia model, the nearest neighbor model, which is very simple in terms of enthalpy and entropy. And we added so this penalty, entropy penalty term, which is also known in this model. And we added these 2 other terms. The reverse loop is the one that happens in the situation what I call the reverse loop. So there is an additional penalty here, and this goes up to the situation where this L is equal to 0, and we have this delta G. Okay, so let's put this everything together. So you have the results here for 4 different origamis. Again, these are rectangular. You have for each one of them, you have the annealing curve and the melting curve. And you can see, so this is the red and the green. You can see that there is a lot of hysteresis in this process. And the dashed line are the result of the model, okay? So you see that we are quite good in reflecting the hysteresis. For some of the origamis, the result is a bit shifted, but in overall, we can say that this catches part of this folding process. Okay, so these are quite old papers now, more than 10 years. And since then, we try to include more information into the description. And particularly, we wanted to look for situations where the fold doesn't happen or doesn't work. So this is a particular origami. So this is a flat triangle. So these are the 3 sides of the triangle, and these are AFM images. So you can see that in many cases, the origami is well folded, but in some cases, you have the opening of one of the sides. So the question is, can we push the algorithm of the description of the folding so that you don't get any triangles, or it's very hard to get triangles? For that, I need to introduce the notion of off-target interaction. So this is part of the folding of this triangle. The green means that the staples are doing their job. Okay, so they are good folded, as you expect, but there are other situations where the staple doesn't do what you expect. So there are 4 different situations. So this one is a staple that is binding to a wrong place along the scaffold, and this is quite common. This other situation is a situation where the scaffold interacts with itself, and this is also quite common. And there are 2 other situations which involve only staples. In this situation 4, you have a secondary structure for the staple, and in situation 3, you have a binding between 2 different staples, which is just a reflection of this type of unexpected interactions. Okay, so I had a quite fruitful interaction with the group of Natalio Krasnogor in Newcastle. And Benjamin at that time was working on this. So he developed a algorithm that for a given origami and given path along the origami is able to tell scaffolds that are not going to give a right folding. How this works? Maybe I should speed up. So again, let's say that we want to fold a triangle. Okay, we are going to sample a pool of different scaffolds. So each scaffold corresponds to a different color here, because the scheme is always the same for the folding. For each scaffold, we need to compute all the staples. Okay, so they are different for each case. And for each scaffold, we are going to quantify how far we are from the ideal model. And for that, we are going to compute the interactions that I called off-target interactions. So this gives you 4 numbers for each scaffold. And the result is this. So here you only have 3 of these measures. Each dot here corresponds to a different scaffold, okay? So this is the energy of the off-target interactions. That means that if this energy is low, you are close to the ideal folding, but if this energy is high, you are far from the ideal folding. Okay, so this is called the Pareto front. This is where solutions are close to ideal, but you also have the reverse front where the solutions are far from ideal. And if you pick scaffolds in both fronts, so these are the structures which are well folded, you expect to be well folded. These are the structures that you expect not to be well folded, and this is intermediate. So the AFM images seem to corroborate the hypothesis that all these 4 measures are telling you something about the folding. And these are the histograms. So deep blue is well folded. And so for this scaffold, it goes up to 60. And for the badly folded, this goes up to 18 only. Okay, so the third question is how these origamis can interact with lipid bilayers. So DNA is perfectly hydrophilic structure. And to convince these structures to interact with lipid bilayers, you need to modify them in some way. And well, the chemistry of DNA is commercially available, it's very easy. So it's very easy to buy a sequence, DNA sequence, which has, for instance, a cholesterol at the end. And this idea has been explored since the very beginning of this field. Here you have 3 different structures that are taken and randomly on this possible DNA organism modified with cholesterol. I guess that the first structure was investigated was this one. Okay, so what you can see in these structures is the presence of a cylindrical part, which is I'm going to call a pore. And the idea behind this is that we expect this structure just to bind to the bilayer and then to create a water channel. Okay. What's interested me in this field is the fact that the number of water channels that you get is really much, much less than the number of nanopores. And I wanted to see why is that? So the experimental setup we have been using is this one. We have 2 water droplets immersed in an oil bath with lipids floating there. You have formation of monolayer around each droplet, and at the intersection you have the formation of bilayer. And when you put on the DNA origamis in one of the droplets, and you do current recording, what you see is the sharp drops, which are really well defined and which correspond to the insertion of a single DNA nanopore. In our case, we studied this DNA origami. So this is basically a rectangle with a nanopore which is crossing across the rectangle. In this particular case, it has a mechanism which can regulate also the length of the nanopore, but I won't discuss here that. These are AFM images, just to show you that this is well formed. And these are the electric recordings we get with these origamis. So I should give some numbers. The concentration here is typically nanomolar or 10 nanomolar. The volume of the droplets is around nanoliters. So you expect around, let's say, 1 million, 10 to the 6 nanopores to be floating and to be able to bind to the bilayer. Still, the number of jumps that you see here is most of the time less than 10, okay? So there are 2 possibilities to explain that. Either the origami doesn't bind to the bilayer or is not able to make the water channel. To exclude the first hypothesis, we did a QCMD experiments where you measure, you first make a bilayer on top of the quartz crystal, and then you measure the mass which is absorbed, which is a way of counting origamis that stick to the bilayer. And as you can see here, this is the shift in frequency. The binding is there. And actually, when you try to make AFM images, which are actually quite difficult because the bilayer is quite floppy, but still you can see patches which look like the DNA origamis. So the conclusion is that these structures bind efficiently to the bilayer, but they are not able to make water channels. So this is a hand drawing. This is a origami which is going to attach to the bilayer. My hypothesis is this is what happens. It binds through the cholesterols here, but the bilayer is very floppy, very flexible. So here there is no water channel formation. In some way to verify that this is something like that, we don't have any real proof that there are these deformations. What we added was a series of positive charges on top, on one end of the origami, and then apply a small electric field to orient. So you create a dipole here to orient the origami perpendicular to the bilayer and to see what happens. And this is my final slide. So these 3, there are no dipoles, and this one has a dipole. So you can see that the number of water channels creations is much greater here although it doesn't reach the number of still we do expect. Okay, so that's it. 3 conclusions. The folding of DNA origamis needs a bit of modification of the usual thermodynamics of DNA to take into account interaction between staples. You can do it in a quite simple way, but if you have kind of nasty scaffolds where the folding process can be trapped, there are chances that you don't get any folding. And finally, this conclusion is more an open question. We know that we suspect that bilayers adapt to the formation, to the binding of DNA origamis. A way to overcome this is to add this dipolar interaction, but there are certainly ways to enhance the formation of these water channels. And with that, I would like to mention these 2 collaborations that I had during this work with Montpellier and Newcastle, the funding, and I thank you very much for your attention.